Action Group webinar. I was going to tell you that the program will be recorded. The webinar will be recorded as it was last time, and it will be put on Aubrey's website. And so we'll give you an update for that. But I'll just share the screen with the actual program, but I thought. Have you got the CRB screen?
Yes. Yep.
Oh, excellent. That's fooling me. Okay. Now, as you all know, the purpose of these webinars is to share experiences and make contacts, and really make contacts in these times, which are pretty difficult for us with the lack of travel and transport that there is for the caused by COVID. And while we're sitting separately, the rhinoceros beetles are still continuing to feed and mate and spread and cause damage. And so we hope that by sharing information, we're going to be able to better coordinate activities and follow up things after the meeting. I'd like to thank all the people that have offered to give presentations and updates today. We're going to do it the same way we've done the previous ones, with a situation update at the start. And Aubrey's going to introduce and coordinate the responses from the Northern Pacific. And then Mark will do the same for the South Pacific. Now, if there are any points of clarification that people want to follow up, or they've got something to add which isn't on the hasn't been in the presentation, please use the function to raise your hand to indicate that you want to talk, and then Aubrey or Mark will stop and let you in to ask the question. After that, we're going to have 5 different science presentations, which each will be 10 minutes. We'll have to keep those short and to time, but I'm sure the presenters have done a good job in giving us a concise format, and hopefully at the end we'll have an open discussion for about 20 minutes. But we will try and keep it to the 2 hours as planned, because otherwise things tend to drift on, and people have the opportunity to follow up with individuals afterwards. Okay, well, I will stop sharing and pass over to Aubrey.
Okay, thanks, Trevor. Half a day from Guam. I'd like to start by thanking Brian Bloss of the UG IT office for setting up this webinar for us. So my job is to coordinate reporting from islands north of the equator. And those islands are starting from the east this time: Oahu in Hawaii, Guam, Rota, which is the next island just north of us, the southern islands belonging to Japan, Okinawa, the Ryukyus, and then Palau, which is also in Micronesia. So because I'm talking right now, I'll just do the quick report for Guam and Rota. And then we'll start from the east, start in Hawaii with Keith Weiser reporting from there. And I'm not sure who's going to report for Japan and Palau. We'll work that out as we get there. So on Guam, we're continuing to see severe damage and mature tree mortality. We're keeping track of things using roadside surveys. We have a very automated roadside survey. We take pictures at a frequency of 1 per second by attaching a cell phone to a car. We drive all the major roads and we come back with 36,000 pictures. And instead of analyzing those by hand, we have object detectors which go in and count V-shaped cuts. And they measure the impact of the rhino beetle on each tree. So we're using Trevor's 5-point damage rating for that. And it's working very well. I forgot to mention that another job that the computer does is it locates every coconut palm in the images. So that's not helping us control the rhino beetle at all. In fact, we are changing tactics right now. We're still focused on finding effective biocontrol for the rhino beetle. It will probably be Rictus rhinoceros nudibvirus, if we can find one that's effective against our biotype. And in order to do that, we need to do lots of bioassays. So we're currently rebuilding our rearing facility, and we're playing around with natural diets. We hope to be able to feed our beetles on ground up dead coconut palms, which are ideal breeding sites for rhino beetles. So it's a very natural food for them. When we get back to our bioassays, we're not just going to concentrate on mortality as the endpoint. We're going to look at sublethal effects, such as reduction in fecundity and maybe reduction in flight capability. So outside the university, there are other people working on rhino beetle on Guam. They're mainly working on surveillance using pheromone traps. And these include Glenn Dulla of the Department of Agriculture. Glenn is also taking down dead coconut palms in the vicinity of ports to try and reduce accidental export of beetles from Guam. And Ken Pulifico is working to help control the beetle on military lands. So they're doing some trapping surveillance on military lands. On the island of Rota, which is just 35 miles north of Guam, they got infested with rhino beetle about 4 or 5 years ago. They are trying to control it, but unfortunately the beetle has not been eradicated and it continues to spread slowly. So that's the end of my report for Guam and Rota. So we'll move 7.5-hour flight to the east to Hawaii, and Keith Weiser is going to take over and report on what's happening in Hawaii. Hey there, Keith.
Yes, I'm here. Sorry, just let me know when you can see the slides.
You can see it.
Okay, great. So I'm going to go through this really quickly, just a couple of minutes. The current status as far as island-wide trapping. So Oahu is still the only island in Hawaii where we've detected coconut rhinoceros beetle. You can see that this is a generally kind of a bad trend. October was a pretty good month though, and we think that was just a correlation, but it correlates with some of the treatments that we've been doing, and I'll get into in later slides. As far as geographic distribution, the original site of infestation is over here, and this is another pretty hot area. These are all the places on the island where we've caught. A lot of the outlying ones are mostly single. So on a heat map here, you can see where the major concentrations of population are in this central portion here. And we've got some spread a little bit northward, which is pretty troubling. But this area used to, down here that I'm highlighting with my cursor used to be quite hot. And we've done about a year ago, we did pesticide injections in there, and this area has gotten much more under control. So the kinds of traditional activities we've been doing are trapping with over 3,100 traps across Oahu, and then some on the other Hawaiian islands on airports. Palm damage surveys, we used to do a lot more of those. Now we mainly do them in heavily impacted areas annually, or in areas where we suspect CRB might have moved to. And then breeding site surveys, we primarily used to do physical perturbation, sifting, and turning of potential breeding material to look for breeding sites. So we've updated some of those protocols. Canines we got a couple of years ago, and they've been trained up, and this year has been sort of a turning point for them, finding a lot more sites, finding in places where humans wouldn't find. So that's been pretty successful. Pesticide injections in host trees we've begun. We got a vacuum steam unit also a couple of years ago that we've been sort of learning how to use. It's been a little bit finicky for us. Very recently, we've been doing fumigation of breeding material, and then we've got some other things in development always. So yeah, as I mentioned, the canines, we have 3 canines, 2 handlers, and a supervisor. And they're basically out 5 days a week looking for breeding sites, sniffing them out. So that's going pretty well now. So fumigation, high throughput treatment of CRB infested material, and eggs have been the most resistant to treatment. So this is work from Mike Melzer that he's been working on. So on electron micrograph, looks like there are some pores that gases could go through in the eggs on these micrographs. So we established some fumigation chambers, so 40-foot shipping containers. One of them has been set up with the piping and the fans and everything to be able to be used for fumigation. So we're able to load it up with a bunch of material. We outsource the fumigation team. Fumigation team treats it and then exhausts it out. Same treatment that we do for termites, like tenting houses. This is in a container. They can also tarp material in roll-off containers and treat it that way. We've done some of that as well. So the test was 128 eggs at different strata in these landscaping bags that are filled with the most resistant seeming mulch material we could find. It's a little more soil-like, very fine material. This was our test. So different CT values, which is concentration over time, which is used for these sort of fumigation treatments. So this is them putting the stuff in those containers. So 18 to 20 hours is when we reach those 2000 CT levels. And then the eggs are then reared out as much as possible and see if there's any survival. So the 2000 CT, the material is completely devitalized, and the controls did survive. So this looks good, looks like an effective treatment. And we've been testing and doing some of these treatments. The 1500 CT was mostly lethal. You can see on that bottom level there were a few eggs that actually did at the end time point reach the larval phase, and we assume that they would eventually become adults with suitable material to live in. Yeah, we got authorization from the manufacturer of the chemical to use the 2000 CT in the field. So this is what we'll use for relatively small breeding sites, and then for larger breeding sites, we just load material into these containers. Then the fumigation company, as you can see on the right panel, will put tarps over there just like they would a house, and then fumigate that material until the sensors tell them they've reached that 2000 CT, and then during the daylight hours they'll haul that material out of the infested area. So that's one of our most recent treatments. We've also been working on host tree injections with imidacloprid. We plan to also include acephate as a 6-month gap filler from the results that we've seen. So the first place we did it was at Iroquois Point, which is that area on the south of Oahu that I showed earlier that we've had some good results. So yeah, we had the mid 20s in average trap finds per week in that area, and after treatment, it was down to maybe 5, and after this graph, it got better. So if you compare the orange graph or the orange dots on the bottom, that's the Iroquois Point injections. So it's a little hard to see on this slide, but Iroquois Point injections, you can see the text at the top. That's when those injections were done, and you can see it dip, and then it rises again, and then dips again. So it's stayed quite low relative. And then we've more recently done Pearl City Peninsula, or PCP here, injections. And we've seen there's a correlation with a drop in fines in that area post-injection. So of course, these are only correlations, but Dr. Sheng has been doing actual research and looking at the efficacy of these treatments. And certainly in the lab, these treatments are effective at the concentrations we expect to be at the trees, and then the concentrations we're reaching in the trees should be lethal concentrations. So, all right, that's the major stuff from Oahu updates. If you guys have any questions, you can email me directly. And then if you want to keep up to date with what's going on in Hawaii, we have an outreach team that you can connect on Instagram, Facebook, or you can go to our website for more information. Thanks. Oops, where's my cursor?
Thanks a lot, Keith. All right, let's move up to Japan. Hopefully Madoka can chime in on what's happening up there.
Yeah, thank you. So my name is Madoka Nakai, and I'm working in Tokyo University of Agriculture and Technology. So in Japan, we don't have a CRB so much as a pest because CRB is not much pest in Okinawa. They don't have a palm tree. So this year, I succeeded the grant to do some more basic science for the CRB. And the question, our questions are whether the CRB G type is a really pest as compared to S type or not. I mean, the feeding activity is more in G type, or of course, resistance against the nudibranchs as well. So that kind of the CRB characteristics we are going to study in this grant. And the second question is, so so whether the CRB density in the Guam is higher, in the Guam, for example, is much higher palm damages as compared to, for example, Palau, is that there are not much, palm damages. So those, the different palm damage places are correlated to the density of the CRB or not. So that kind of density, analyze, we're going to do it. That is another question. The third question is, whether the CRB G type, which infected with nudibranchs, reduce the feeding activities or different sublethal effect that's going to be studied. So those three questions we try to answer in this grant. So in this grant is international and supported from Japan, Japanese Society for Promotion of Science, and involved that Avery Moore and Sean Marshall and Skevan Eteberry and Michael Furon. So that, the Australia, America, and New Zealand, and Japan collaboration. That's all. Thank you very much. Thank you.
Thank you, Madoka. Unfortunately, I don't see Chris Kittelong from Palau here. So I think we're going to skip Palau for these reports. And that basically concludes what's happening north of the equator. And I guess we'll move down south now. So who's doing that? Trevor, over to you.
Okay. Mark, are you ready to present that? Mark, I can see you, can you hear me? You're muted at the moment. Okay, I don't think we can get Mark. So one of the things, Sulav, do you want to, can you, who else do we have on the list? Bala is going to present later on from Papua New Guinea on damage assessment, and perhaps she can update us on any points. Mark, are you with us? But going back to the program, and Sulav is going to present on the position in yes, on the position in the Solomon Islands. Mark's having trouble with the audio, so he's going to go out and come log back in. I think probably our best idea is to go on with the science presentations and then come back to the situation updates in the discussion at the end. So I'm very pleased with, we've got five different science presentations, and I'm very pleased that Jeff Bedford has agreed to kick off for us and talk about his experiences in the earlier waves in in Fiji and Papua New Guinea. So Jeff, are you able to turn your microphone on? At the bottom left of your screen, it should be. It's the in the square for the Zoom webinar. There should be a taskbar along the lower left-hand end.
Can you hear me now?
Yes, we've got you. That's good, Jeff.
Right. I had a bit of difficulty at the last minute in trying to send the PowerPoint to Sulav. I got a message on the screen saying the PowerPoint attachment presentation exceeded the allowable limit. So I was trying to split it, but I ran out of time. So to get the PowerPoint on the screen now,
go to share screen at the bottom in green in the middle.
Yes. Yes, I've got that, and I just
click on the screen.
Yes.
To share.
Well, I've clicked on screen.
Yeah, I think it's at the top, isn't it? That there's a thing that says share screen.
Yeah, click on screen share, and then in that same box on the right on the bottom, you have to click on share.
Yeah, so share screen. Yeah.
And then choose your screen. It should say screen, whiteboard, or other programs you might have open.
Well, I've got the PowerPoint in my documents.
Can you try opening the PowerPoint first? And you can also just share your PowerPoint if that's what you want.
Right. I see. Yes, I'll go into my documents. I've gone into my documents, and
there you go. You're sharing your screen.
Yeah.
We yeah, just locate your PDF that you want to share.
The pictures. The pictures are blocking.
What's the name of the?
The one I want is over here where my picture is.
Oh, do you see on top of the pictures? You can actually just drag, like just left click on top of the pictures, and you can drag the videos out of the way.
I think I've got it. I've got it now. I can click on it now.
All right, go ahead and open it.
I've got it in front of me now. So
That seems to be coming up.
so you can see it at your end.
Yes, we can see your actual documents folder.
All right. Okay. All right. Well, I'll start going through it now. I was involved with the release of Ehrlich's nudibranchs against coconut rhinoceros beetle in Fiji in the 1970s.
Sorry, Jeff, we can't actually see the slides yet.
Oh,
Yeah, can you minimize your documents folder?
yes. I'll minimize this. What I'm showing now.
The documents folder, yeah.
Am I? Are the slides showing up?
No, not yet. Can you see them on your screen?
No.
I was minimizing them.
Do you have multiple screens, sir?
I've got the PowerPoint has now come back onto my screen. It says your screen sharing is paused. Do you have a suggestion as to how I can?
Okay, I'm gonna stop the sharing, and can you try sharing again? Just try sharing the PowerPoint itself. Okay, so I stopped your sharing. Try pressing share screen again.
Yes.
And just choose the PowerPoint.
Yes, that's here. Yes.
All right, so click on that and then click on the blue button on the bottom right that says share.
Okay, I'll click on earlier, right, and share, right? Okay.
Yes.
Yes.
There you go, and then you can start
Yes.
your PowerPoint show.
Oh.
Yes, we can all
Well
see
done,
it.
Jeff.
Okay, shall I start now?
Yes, yep. Thanks,
Okay.
Jeff. Well done.
I was involved in release of Ehrlich's nudibranchs against coconut rhinoceros beetle in Fiji in the 1970s. Virus was produced by having field collected larvae feed on virus contaminated food. They died of the virus disease. The dead larvae contained much fresh virus material. Virus was initially released into breeding sites. Later, we moved to infecting adults and releasing them to spread the disease into the wild beetle population. This required a steady supply of adults, so a mass rearing facility was set up to produce adult rhinoceros beetles from the egg stage. A batch of adults produced from the mass rearing. Adults were infected by being immersed in a crude virus suspension made by ground up virus grubs mixed with water. Beetles were immersed in that. Their mouthparts were contaminated and they were thus infected with the virus. Infected adults being released. Two types of damage survey were used as an index of changes in the beetle population. The rapid damage survey measured the number of palms with damage to central fronds in a sample of palms. Detailed damage survey measured the amount of damage to fronds in a sample of palms at a particular location. 3 locations on Beetle level are shown here. One was along the road between Nandi and Lautoka on Viti Levu. Another location was at Thamboni on the north coast. Third location was at Tamavua near Suva. At the start of the virus release, damage was at a high level in palms at those locations. In the years following the establishment of virus, damage fell to much lower levels, such as shown here. I left Fiji in 1976, but was able to revisit the sites 35 years later in 2010, and found that damage at those sites was still at relatively low levels. And one of the sites, Thamboni, it had fallen even a little further. During the work in Fiji, damage fell at a number of other virus release islands and locations, as has been documented in literature at the time. This shows a group of palms at a location on the north coast of Viti Levu, just before virus release and establishment, showing considerable damage to the palms at that location. Then after the virus became established, the palms at that location showed a considerable improvement. Just some comments relevant to the situation then and to today. The higher the damage at the start, the more noticeable is the fall off in damage after the virus becomes established. The virus used at that time was freshly produced from larvae it had just killed. Every patch of palms or palm location is a different palm beetle virus ecosystem, in the sense that each differs from one another in many different variables. The virus does not make the Rhinoceros beetle disappear. The beetle is still out there everywhere, so palm damage still goes on everywhere, but apparently due to the virus, less. So people have to get used to a certain level of damage still continuing on. Since that time, much of the virus released in various specific locations over the past 4 decades has been produced in tissue culture of the African black lawn beetle, Heteronicus aurata. And in tissue culture, the replication cycle, that is the time taken for the virus to reproduce in the cells, is about 16 hours. So if it has been in culture from 1981 up to the present time, many thousands of replication cycles may have occurred in producing batches of virus. So is there a possibility virus isolates may have adapted and evolved by mutation to culture in Heteronicus cells and may have lost or had a reduction in their ability to infect Rhinoceros beetle cells, as the Rhinoceros beetle is their intended target, but in tissue culture, they never come into contact with it. The virus has plasticity in its genome. When it was released in the Maldive Islands, Indian Ocean, genomes appeared in the field within years of its original release. So viruses are well able to mutate, for example, the current COVID situation, and also flu viruses. I would suggest one could fully sequence the genome of Heteronicus cultured virus isolates, and that means the sequence the whole genome, not just parts, because mutations could be hiding anywhere in the whole genome. One could also sequence genome of various field collected virus isolates coming from different geographic locations, then compare them and look for genome differences. And if any differences due to mutations are found, one could test field collected virus isolates for their infectivity against Rhinoceros beetle larvae and adults. If releases of existing virus isolates or newly collected isolates are planned into areas where virus is already present, I would suggest one could collect from the field and hold individually samples of Rhinoceros beetle larvae and adults to see if disease develops in them. This would give an idea of the incidence of already present virus at those locations. And it will also give an idea of the incidence of endemic pre-existing fungal pathogen Metarhizium anisopliae if release of Metarhizium was also being considered in those locations. As the virus and Metarhizium compete or conflict with each other to infect the Rhinoceros beetle. So mortality due to one leaves fewer hosts for the other pathogen to multiply in. And lastly, I've noted in recent literature a Rhinoceros beetle biotype or haplotype previously thought resistant to virus has now been found to be infected with the virus at 2 locations based on studies of samples from the Solomon Islands by Dr. Itabari and based on samples from Palau, based on work by Dr. Tanaka and Dr. Madoka and colleagues. Thank you.
Right, thank you, Jeff. That's very good. It's great to see your experiences with that wave of rhino beetle in Fiji. And you've raised a lot of points which I think you'll be interested. Some of those are going to come up in the rest of the meeting, but and we'll get back to those in the discussion. But if anybody's got any specific questions on Jeff's experiences in that field release, we've got time for 1 or 2 quick questions now. And because I've got the floor, I'll start with the first one. How many infected beetles did you release, Jeff? Do you have a feel for how many it's necessary? Because in some cases, a lot of beetles have been released, and in some cases, it seems like very few have been released with the virus, and it's allowed spread.
Offhand, I couldn't give any actual figures of beetles released. I'd have to look that up in various records that I have.
But it would be quite a few.
Yes, but the numbers of beetles released it would vary depending on the supply of beetles. So to improve this supply, was why a mass rearing facility was set up, but the actual number of infected beetles released at a particular location would vary, and it would depend on the number of beetles that were available at that particular time. But I would need to look up records that I have if actual figures were needed.
Okay, thanks, Jeff. Were there any questions from participants? I'm not sure that I can see. We'll probably come back to these in the discussion time anyway. So thank you for Jeff for that. Can you stop viewing your screen, sharing your screen now?
What's the procedure for that, Trevor?
We're right.
I just click share screen.
Yep, we're okay now. That's good. Okay.
Right.
One of the things we've been looking at, working with our colleagues, is pretty much a big investigation of the situation in the Solomon Islands. And we've been putting that together from reports and the investigations going on. And Sulav has got a summary of where we're up to with the situation in the Solomon Islands. Sulav works for AgResearch and has been with us for a little over a year now, isn't it, Sulav? But
Yep.
I'll hand over to Sulav.
Sure. Okay. Are you able to see my screen?
Yes, that's good.
Okay. Thank you, Trevor.
Sulav, you've got the slideshow screen, not the full screen.
Can you see it now?
That's it. Yep.
Okay. Thank you, Trevor, and thanks everyone for joining in today. So yeah, as Trevor just mentioned, I'm just gonna give a brief summary about our work in Solomon Island with coconut rhinoceros beetle regarding its spread and distribution across the country over the last 6 to 7 years. Let me first acknowledge our team members: Sarah Mansfield, Nikki Richards, Miriam Tubolsev, Trevor Jackson, and Sean Marshall from AgResearch. Similarly, Francis, Crispus, and Gideon from the Ministry of Agriculture and Livestock in Solomon Island. So just a brief background on coconut rhinoceros beetle in Solomon. I think we all are quite aware that CRB, in short, coconut rhinoceros beetle, is one of the major pests of oil and coconut palm globally, and it was detected. It was 2007 when the new wave of CRB invasion was detected from Guam, and then subsequently from surrounding island countries and territories. And in 2015, it was confirmed from Honiara, the capital in Solomon Island, and the team from AgResearch and other collaborators, they found that the new wave of invasion into the Pacific was because of a previously unrecognized CRB haplotype called as CRBZ, which was apparently tolerant to the commonly used biological control agent, the virus Odysseus norovirus, in short ORNB. So a quick note on methodology. Actually, after CRB was confirmed from Honiara in 2015, the local Ministry of Agriculture and Livestock, in collaboration with different regional and international partners, they started this massive awareness campaign to educate the public, the general public, farmers, extension officers, biosecurity officer, and other stakeholders regarding CRB and the threat it poses. So what happened was basically once any reports, any field reports of CRB like damage or any sightings of CRB adults or larvae, the local MAL team, the Ministry of Agriculture and Livestock team, they conducted a series of delimiting surveys and exploratory field visits to basically to determine the boundary of CRB infestation. And at the meantime, they also collected CRB samples, both adult and larvae, as you can see here in the picture in the right. And those samples which were collected during those field visits and exploratory surveys, they were sent to us here at Ag Research, where we conducted genetic analysis to determine haplotype as well as presence or absence of the biological control agent ORNB. And like the results from these genetic analysis, along with the anecdotal reports from the field, were used to basically were analyzed and used to basically track the invasion pathways of CRB within Solomon Island, and in the meantime provide frequent recommendations to the local ministry regarding appropriate and tailored biosecurity measures based on specific outbreaks within certain region or certain island. So next, I'll just quickly run through the timeline of CRB within Solomon Island, just to give you an idea about like how fast CRB has been spreading across Solomon Island. So as I mentioned earlier, it was first confirmed from Honiara, the capital, in 2015, and then in 2016 it was confirmed from Shetland Islands in the Western Province. Then in 2018 from Rhinoceros Island in the south, as well as one tiny island in the Western Province, Gijo. In 19 from a bunch of places in the Central Solomon, it is confirmed from Ulawa Island, as well as a couple of islands in Malaita, as well as islands from Central Province. In 2020, it was confirmed from a couple of islands in. I will come couple of islands in the easternmost province of Temutu, as well as in Choiseul in the west. Similarly, it was also confirmed from like one of the very remote northern island around here, called as Java Atoll in 2020, as well as confirmed from Isabel province. More recently, it has been detected in Makira Island as well, in a place called Kirakila. Actually, we have just received the samples from Makira, so yeah, analysis is ongoing. And when we conducted, when we tested these samples for haplotype analysis, we detected 2 distinct invasions. So the samples which are collected from Honiara, around Honiara, they were determined to be CRBG haplotypes, and the samples of which matches closely with those from the new recent wave of invasion into the Pacific. For example, those collected from areas like Guam, Hawaii, as well as mainland PNG. Whereas CRBS haplotypes were confirmed from the samples collected in the western region, from the Shetlands and the western provinces. So, and these samples actually matches closely to those collected from the outer island of PNG, which represent earlier wave of invasion, as well as from the like the center of origin in the Southeast Asia, South and Southeast Asia. So yeah, here is the current distribution of CRB based on all the reports that we have, and separated by 2 different haplotypes, CRBG versus CRBS. So as you can see, that CRB is now well established in areas around Guadalcanal, also like the northwestern corner of Malaita, and islands in the central provinces. And there are already reports of them being highly damaging to the palm. And so apart from these places, the other outbreaks are still like small and isolated, and we haven't been able to find a high number of trap catches so far. So which basically suggests that like it is much more likely that these are much newer infestation or much like recent outbreaks. The other thing regarding like the 2 haplotype distribution is, as you can see, like the CRBZ is like dominant in the central Solomon as well as the eastern region, whereas CRBS is highly dominant in the northwestern corner of the whole country. We have been also like monitoring the presence or absence of the biological control agent ORNB in all these samples, and it was very recently. In fact, it was only in 2019 when the virus was detected from samples collected from this area in Guadalcanal, northeastern region. So So it was in 2019, and more recently, it was also like confirmed from one site in Malaita. Apart from this, like all the rest of the population are still virus free, and we haven't been able to confirm any virus from other populations. And overall, we have analyzed around 400 CRB samples throughout Solomon Islands, and out of those 400 CRB samples, almost 65% of those samples are confirmed as CRBG. And as I mentioned earlier, all these samples closely matches with those of the recent wave of invasion into the Pacific. Similarly, the rest of 35% of those samples are confirmed as CRBS, and which matches again closely with those from the outer island of PNG as well as from the center of origin. And out of those 139, we have found a minor one SNP variation in eight of the samples coming from a Choiseul, and a couple of from Southern Islands. So those are very recent minor variation that we have found so far. So with that, just to give you a summary, as I mentioned, like we have kind of identified two distinct invasions of CRB has been identified. One starting from Honiara in Guadalcanal, whereas the other one from the Southern Island in the Western Province. And the two different haplotypes has been determined, CRBZ and CRBS, and which are spreading quickly throughout Solomon Island. Mostly, I think most likely assisted through human transport in forms of transport of goods or services, goods or supplies. And overall, we have found that like the haplotype analysis or the genetic analysis of the samples that we have conducted are basically consistent with the local observation of the pattern of the spread. So yeah, with that. Let me thank our funding agency, New Zealand Foreign Affairs and Trade, and our local implementing partner, Ministry of Agriculture and Livestock, Solomon Island, as well as Biosecurity, Solomon Island. Yeah, that's all I have.
Very good. Thank you, Sulabh. Any questions for clarification from Sulabh? Okay. This is Madoka.
Thank you for a very interesting presentation. So I may miss that information, but did you talk about how was the palm damage, among that, the island? Because it will be S type or G type, different type, and virus occurrence, whether it is, you have any idea whether the virus existence island may be, lower palm damage, or can you explain about, concern, can you mention about, so whether the palm damage will be correlated to the virus occurrence and at the same times, the genotype or haplotype, differences?
Yeah, I think in one of my slides, if you see one of my slides, I can't share my screen for now because somebody else. Can you stop? Okay, let me go back to my slide. Where it is here. Okay. Okay, so yeah. So if you see my slide here, so basically, so you can see, like the CRB is like quite widely established in these areas around, and most of these samples from these areas are CRBG. So far, like looking at it, we have found that CRBG has is more damaging than CRBS. This is just based on the observation so far. So yeah, but again, like very few percentage of those CRB samples which have been collected from this area are with virus. So we haven't seen much virus in these areas so far. So yeah, that is kind of basic observation that we have so far. Yeah.
Yeah. So the palm damage, the coconut, damage or oil palm damages are less in that S-type, only S-type existing short island. You said, north part is a less palm damage or not?
Yeah, actually, that's what we have found. In fact, there was a recent survey done in, this Gizo Island. In Gizo, there has been CRB for almost now. I think it was confirmed in 2018, and it's been there for 3 or 4 years. And there was a very recent survey done, and we didn't found, like much damage so far. So yeah, that's our expect. That's what we have seen so far. Yeah, much less damage with CRBS population.
Thank you. I want to ask more about it later, but I want to know how
Okay.
much. How did you have a density survey did you do? But I will ask again. Thank you very much.
Sure.
Okay.
Sure. Thanks a lot.
Come back to you later, Madoka, but Jeff, have you got a quick question?
It's just a quick question. So based on the information that's presented, the virus is well able to attack CRBG as well as CRBS in the Solomon Islands. So does that mean classifying beetles according to haplotype and based on their mitochondria is not so relevant now, since both types of beetles are able to be infected and with the virus and carry it? So does that mean that the haplotype classification of beetles is less relevant now based on this new information?
Okay, so I think, Jeff, I think that is not my forte. So maybe I'll ask my co-author, his son, to maybe Son or Trevor, if you can, like kind of respond to it. Yeah. I
think what we'll do, Jeff, is come back to this in the discussion, because this is a controversial point, and it requires more time to actually discuss it. But I can say shortly that we have not yet found virus infection in pure G populations. Once the two haplotypes are together, there is the opportunity for crossbreeding. And so the haplotype no longer follows the nuclear genes, which can give resistance. And so we can see from that the possible effects of mixing. A G haplotype from a mixed population may still be sensitive to virus. And the other thing, resistance does not necessarily mean total immunity from disease. It means that the disease is less able to infect and multiply within that organism. So there are a few complications that we'll come back to in the discussion, perhaps. Okay.
Yep.
Sorry, we've got to keep moving. I think Mark was ready to come back to us with a bit of an update from Papua New Guinea, and that will go on quite nicely into Bala's presentation. So Mark, are you there? We can't hear you, Mark. We can see the screen.
I think Mark is on mute.
Hello, can you hear me now?
We've got you, yes.
Sorry about that. My computer is playing a bit for now. I can hear you all clearly, but I can't. You can't hear me. So there's probably something wrong with the audio that we'll try and sort it out. But thank you for the time to share the information for the South. I'll be using Celeste's computer for now, and then I'll get back to my computer after this. But my update will be very brief. I'll just present for Vanuatu and PNG. For Solomon Islands, Sulav has already done that in detail. And yeah, for New Caledonia, we have not received any information, so I can't provide any update on that. But I'll just provide for PNG and Vanuatu for now. Okay, I'll run through that quickly. So that's the situation for PNG. If there's any one from PNG that's attending and would like to provide any comments or any additional information, they can be able to do that. But this the map that's showing the distribution of the beetle within PNG. So the CRBG, it still remains on the mainland. Initially, it was in Port Moresby. Just where the green dot is, that's where it was detected, and that's NCD, Port Moresby, and then it spread along the eastern coast, going towards Milne Bay on the other side, within Central Province, and then it's gone the west coast up to the Gulf Province as well. So it's gone into the Gulf Province, and then it's still moving into the Central Province towards Milne Bay. And more recently, last year, I think 2019, it was also confirmed from Medang on the other side of the mainland, and it's confined there. And they in Medan, they also have a mixture of both the CRBG as well as the CRBS. And CRBS has got virus infection, but all the population of CRBG in PNG are free of virus. And the New Guinea Islands, all of it is CRBS. That's including the New Britain, West and East New Britain, New Ireland, as well as Manus Islands up at the north. And we also have some CRB S on the border of Solomon Islands, bordering with the Western Province that Sulав has highlighted. So, and for those population, they also think without the virus. But at the moment, both KIK, PNG KIK, Nakia, as well as PNG OPRA are all involved in the awareness and management programs within the country. So that's they're looking at the awareness materials with some support from us that they're distributing, and also the training programs, and then looking at management efforts within the respective areas where we have the indication of the beetle. So that's the situation in PNG. And for Vanuatu, thanks to Armstrong. Armstrong, if you have any information, I think you are joining in as well. So he's provided the update. You're joining in as well. So if you have any information that you would like to contribute, you can do that. But for the time being, it's mainly concentrated on Efate, which is the main province, where the main provincial, not provincial, sorry, the country headquarters is. So it's mainly concentrated on the western side of the island. And the initial detection was at Magaliliu, just somewhere there in 2019. And then it's sort of, it's been spreading, and it's gone on to the adjacent islands as well. That has been confirmed. And it is still CRB as at the moment. And the ministry has sort of put in some restrictions, particularly with the movement of boats within the country. So they've imposed a ministerial order for the boats to try and leave before dusk, and also if for the boats that are living in the a little bit into the night, they have to dim their lights before they leave. So there is some enforcement of quarantine on the movement, and there's also inspection on the boats that the biosecurity team is doing. The main concern is getting them onto the other islands, including Santos, I think, which is the main coconut producing province within the country. So that's what they're trying to do, and they're also looking at the meteorism application, I think, as part of the management field sanitation, and then also have done some surveillance and monitoring, and also involving awareness program within the country. So that's basically for Vanuatu, and otherwise, as I've indicated, Sulawesi is updated for New Caledonia, for Solomon Islands, and New Caledonia, we don't have any information to provide. And that's it for the Southern Pacific. Thank you, Trevor.
A quick question, what did they taste like?
They were okay. I think what's his name? Benoit is also in the one as well. The dish to the left or to the right, I think, is, and one of them is mine. And I think I lasted for only 5, and Benoit had a bit more than me, about 10 or something. But it was. It tasted more like a plastic, so we were uncomfortable with it. We just wanted to try it ourselves before we try and encourage other people to have it, but it wasn't comfortable. Yeah.
Okay, so humans as a predator doesn't seem to be the best option.
That's right, I think.
Okay. Okay, do we have a quick question for Mark at all? We'll come back to it in the discussion. Okay, we're going to Mark. Can you turn off your screen? Because we'll move next to Bala from Nakia from PNG, and Bala has recently returned to PNG after completing her master's degree down here at Lincoln University. And has stepped back into the battle against CRB. So we're looking forward to her presentation. That's good.
Hi,
That's
everyone. Yeah, thanks, Sulab. So hi, everyone. Today, I'm going to be sharing results from my master's project. As Trevor mentioned, this was one I undertook at Lincoln under the supervision of Sarah Mansfield, Mike Bowie, and Trevor Jackson. In the project, I investigated the methods used to measure damage caused by coconut rhinoceros beetle, CRB. Next, please. So damage assessments are essential in that they determine if the beetle is present or absent, or if the palm damage from CRB is increasing or decreasing. This provides an indication whether control measures are helping to reduce the pest numbers, or if the populations of this invasive pest are actually growing. From reviewed literature, there is no consistent method for damage assessment described. In fact, each researcher had developed their own approach. And much of the work done on the pest dates back to previous invasions and were developed at a time where digital cameras were not readily accessible. Also, there has been no comparative studies of the existing damage assessment methods to indicate which approach is easier to use or which is more accurate. Having said that, the work I'll present today address the issues of accuracy and consistency between observers, whether two people using the same method can reach the same conclusion for a given palm tree, or does experience, having worked with coconuts or coconut pests, improve accuracy. Next, thanks. So 3 damage scales were used that increased in complexity. The first and simplest was the binary scale, which had options of undamaged and damaged palms. The next was the 3-point scale with undamaged, damaged, and dead categories. The most complex one was the 5-point scale that consisted of undamaged, light, medium, high, and dead categories. So this data was gathered through an online questionnaire that was administered through various professional networks. It's probably likely that some of you had participated in this survey as well. So this method was used to test observer accuracy. Participants were presented with a set of 20 photos to assess in a randomized order. These photos were pre-selected by a panel so that 4 photos represented each damage category from the 5-point scale. Participants were required to assess all 20 palms using the binary scale first, then followed by the 3-point scale, and finally with the 5-point scale. In the letter section of the questionnaire, we asked participants to provide some demographic information and about their prior experience with CRB. So to be honest, I was a bit nervous not knowing what the feedback would be like after the link was sent out, but it turned out that the response to the survey was great. We had 241 completed surveys, which gave us a great data set to work with. Overall, the pool of respondents was quite well balanced in terms of gender and age group. And nearly 50% of the respondents were scientists. Now, this was expected considering that we had circulated the survey through the professional network or a science-based mail system. And then people residing in Australia and New Zealand had made a bulk of the population for the respondents' population, that is, so followed by residents from the Pacific, and nearly 3/4 of the respondents were not experienced with CRB. In discussing the results, a correct answer meant that respondents placed the palm in the appropriate category according to the panel's designation of the pre-selected photos. So you can see here that there were more wrong answers, indicated with the dark bars, as respondents moved from the binary scale to the 3-point and then onto the 5-point scale, which was more complex. We were surprised to discover that while most people got the palms correct using the 5-point scale, no one had scored all 20 palms correctly in this scale. But for the simpler scales, binary and 3-point, some respondents did classify the entire set of 20 palms correctly. Next. Okay, got it. From assessing the effect of experience on response quality, we found that experience did improve performance because this group of respondents were more likely to classify PAMs correctly. What was also noted was that even for experienced observers, accuracy still declined as the damage categories increased from binary to the 5-point scale, leading to more incorrect responses. To investigate further, responses were broken down in relation to each PAM. Okay, this first graph shows responses for the 4 undamaged PAMs using the 5-point scale. The arrows mark correct answers, and you can see that the majority of the respondents had categorized these PAMs correctly. Next. Thanks. So this graph looks at the 4 PAMs from damage category 3 in the 5-point scale, or the medium damage in category 3. Now you can see that there's a greater spread of responses. However, most people failed to classify the PAMs correctly here. So we had majority of respondents classifying or categorizing these PAMs as category 2 instead of the category 3. So the medium category was the most difficult for respondents, followed by the low damage category. PAMs that were severely damaged or dead were mostly classified correctly, suggesting that the extremes of damage were easier to assess. So in conclusion, the first point we noted was that training is essential to improve accuracy. In that way, trainees can learn from experienced observers, but training is not the whole answer because even experienced observers were seen to struggle with a 5-point scale. However, only the 5-point scale has the capability to detect the early stages of PAM recovery, because the scale can pick up signs of recovery from severe to medium to low damage. During the course of the study, some preliminary investigation done suggests a trade-off between ease of use and statistical power. And we plan that in the future to do a more in-depth study around this aspect. And yeah, I think that's it. Thank you.
Thank you, Bala. That was very well presented. Oops, I've got an echo somewhere. But no, very, very good presentation. We've got time for a quick question to Bala. Perhaps we'll follow up in the discussion. Okay, well, we'll move from the field and the practical and the Beetle on the move to some of the science it's harder to see. And James Grosella from University of Guam is going to present on developing well, James, you tell us what you're going to do. Are you there? Can anybody see James?
Actually, I've just promoted him as a panelist, so he might be able to share his screen now.
Ah.
So yeah, hopefully, yeah.
Into the unknown. Yep.
He's muted though. You know.
Oh, okay. Trevor, can you hear me?
Oh yeah.
Yes, we can hear you now, James.
I'm sorry. This is first time. A little rusty here on this thing here. So how do I get my PowerPoint up? Is it up?
Share screen.
Okay.
And then you have to select your PowerPoint, and then you have to press something else.
All right, let's see. Where's the share screen?
Your screen's at the bottom in the middle.
Okay. Chat, I see chat.
This is Aubrey. I
Oh,
don't see Jim as a panelist. He's still in the attendees box. And I don't think the attendees are able to share their screens. Brian, are you there? Can you help us? I tried to promote Jim Grisella to a panelist and it didn't go through.
Oh, is that what that was? Oh, geez.
Oh, okay. Did you undo it? You end up in the book. Okay, I'm going to try it again, Jim.
I was trying to advertise something.
Put you over to a panelist. Promote to panelist.
All right, join it. Okay, here we go. I got it. Okay, join.
Okay, it's yeah. Now you're a panelist. You've got superpowers, and there should be a share screen button at the bottom of your page. Green button. And also you need to unmute yourself. Okay, we can see your screen, but it's not your PowerPoint. There we are. Fantastic. Good job. Jim, we can't hear you. Make sure you're unmuted. Jim, if you can hear me, there should be a microphone at the bottom left-hand corner of your screen. And you need to click on that.
Okay.
We can hear you and we can see your shared screen, but we're not. There's your PowerPoint.
Here we are. Yep.
You got it?
Yeah, you're all set.
All right.
To slideshow view.
Sorry about that. I was a little.
Go to slideshow view, Jim.
What's that?
Go to slideshow view for the full screen.
Okay,
Along the top.
I got it. How's that?
Terrific.
All right. I was a little hesitant to present this seminar, the talk, because I don't have a lot of data, but I thought I'd get it out there. So anyway, this is the title of what I'm going to talk about. As you know, there's been a lot of talk about trying to establish the cell line from the CRB, and these are some of the beetles that have been established in cell culture, except for this guy down here. I'm not sure about this one. So I thought, well, maybe, I have some experience in cell culture work, so I thought I'd give it a try. It's something that we could use in comparison with what we already have. And I think it should be produced from the insect that's where the virus came from. We may get a better handle on what's going on. This is what I've come up with in terms of trying to extract tissue and cells it's pretty detailed. It's just a lot of manipulation, and the concern is that I try to avoid contamination. That's the biggest problem. And the last couple of weeks, I was lucky enough to get something going without bacteria or fungus growing in the cells. I'm not going to go over it. But anyway, this is what I've come up with so far. This is pretty early. This is cells grown in high clone. It's a commercial media that's produced for insect cells. And I have I've added like 10% of field bovine serum to the media, which seems to help cell culture. You see clusters of tissues attached here. And here's a little bit later on in the same cell culture media. You can see the cluster of cells there. This is tissue attachment, and eventually, hopefully, the cells will start to emerge and spread out, start really replicating in the media. There's another picture. This is another serum-free media. I don't have FBS in this cell culture. This is a media that was actually produced for mammalian cells, and this is something I tried using it to generate marine sponge cell culture, but it never worked. But here, it seems like it's doing something. You can see the individual cells there, and whether it'll take off, that's a question of a little bit of time. This is what I've done in terms of looking at different media, and I've tried different sources of tissue cells, the embryo and gut and the hemolyth. The gut's going to be a real problem because it's got a lot of material in it that needs to be cleaned out before I can actually work on it. Embryo has been my best hope, and that's where all those pictures have come from, and embryos. This is the different cell media. This is another commercial cell line that's developed for insect cells, and I've gotten some cells growing in that. Some of these I haven't tested yet, and I have tested some, but again, contamination. So I'm still working on that. Let's see what else. So if we're able to get a cell line going, and that means that the cells become confluent and you can pass it in culture. In other words, if you take the cells that are confluent in a flask, and you split them up and you pass them to a new fresh flask, and you can keep doing that until you get about 7 or 8 passages. And that number's not etched in stone. It's something I've dealt with, and it's from my experience. So once that is accomplished, then we need to do a bunch of other things, which I've listed here. And the first thing that's really important is to compare your DNA from that cell line with the DNA from adults and larvae. Make sure that you confirm that your origin of the cell is same is what you got. And it's important also to keep track of passage number. A lot of people don't indicate what passage number they're working at when they're working with cell lines. So you can kind of keep track of that. And another thing is, once it's established, it's important to preserve the cells at an early passage, because later on things may change and the cells may die out or something may happen. Who knows? Then later on, you can work on determining cell growth of the line. You do a karyotype of the cells. You have to evaluate for mycoplasma contamination. That's important. And then you can do some more detailed studies of infectivity of the different CRB virus isolates in cell line. And I like to use TCIV50 because it actually measures infectivity, as opposed to, say, using qPCR, which only really measures number of virus particles. Doesn't tell you how efficacious the virus isolate is. Anyway, this is kind of where I'm at. And this is all I've got. This is my story for now. So I appreciate your attention. Thank you.
Okay, thank you, Jim. I'm sure that's a lot more complicated than you know, the few results that you can present, because it's very difficult to get the cell lines established without the contamination.
Yes,
Does anybody have a point or a question for Jim? Two. I've got. Jeff Bedford.
Yes, sir. I would just comment that it sounds very encouraging to explore cell culture of cells of Erectes rhinoceros, as that's the intended target organism, particularly for the Erectes nudibvirus.
Yes, I mean, it's something that probably should have been done years ago. I mean, when I worked for USDA quite a few years ago, we had one of the isolates from CRB, the virus, and I wanted to work on it, but we didn't have the cell line, and I was told by powers to be that that wasn't our mission, so I never was able to do anything with it. So I think it'll probably give us a lot of new information. Certainly a lot of comparative studies that can be done. And, we'll see what happens, especially the genetics of the virus. That's something.
Yep. No, very good. Very good, Jim. Sean, have you got a comment? I see your hands up. But your microphone isn't. So, okay, we'll keep moving and come back to that. Another microphone problem. That seems to be the problem of the day. We're going to move on to our last formal presentation, and that's Jean Jacobs from AgResearch is going to give an overview on genetic analyses that are being carried out at Lincoln. So Jan, over to you.
Let me see this screen.
We can hear you.
To see us. You're going to see yourselves for a little bit while I get this up. Right. And I probably have to display setting. Now you should be able to see the screen. Is that correct?
Yep, that's good.
Okay, let's start here. Thank you for the invitation to be able to present our work here. That I'm the one that has to. I'll share my video too. That's probably a bit more sympathetic. Sorry about that. I'm part of a big team, and I have been given the honor to do the presentation here. So what I'd like to talk to you about today is the genetic analysis for improved decision-making and CRB management. And that consists of, slides are not moving. Next slide. No, slides are not moving. Why are they not moving? Here we go. So it consists of 3 different organisms that I'd like to highlight today. It's not just the beetle that we're all trying to tackle, but it's also 2 of the biocontrol agents. One is the virus, Erectes rhinoceros nudibvirus, and the other one is a fungus, Metarhizium majus. So I'll be discussing some bits and pieces of work that we've been doing in the genetics and some genomic work of all 3 of these species. While I get this sorted. So you would have seen this picture, most of you would have seen this picture out of a presentation, out of a publication, I should say, in 2017 by Sean Marshall and co. And it is based on a gene sequence of a CO1 gene in the mitochondrion of beetles. And it largely distinguishes the geography of where the beetles were collected, and it also distinguishes virus tolerant versus virus resistant. Now, there have been several iterations of this figure and different ways of presenting these data, but all in all, when we look at full sequences of the mitochondrial genomes, and in that case, we have used our own sequences that we have produced, as well as public domain available mitochondrial sequences, it gives the same outcome. Basically, what the CO1 gene clade definition puts up is the same when we look at the wider mitochondrial genome. So the CRB genomic variation, when we look at the whole genome, why would we do this? Well, it's to understand the variation at a larger scale, just that one gene in the mitochondrion. And it will help us to develop robust identification tools going forward, because I think Jeff already mentioned that we can distinguish different clades of CRB using the CO1 gene, but over time we want something probably a bit more robust to distinguish the different geographies and etc. So we have generated whole genome sequences from beetles, and we are in progress of doing more. We have completed 2 out of Guam, and they belong to clade 1. 2 from the Solomon Islands, they belong to clade 1 as well. 1 from Vanuatu, belonging to clade 2, and we have 1 more in progress, which is out of the native range in Malaysia. So why would we do that many different genomes? Well, we would like to get nuclear genome-based markers for detecting CRB and to determine the geographic origin of the CRB that we have in hand. So it might be collected in one place, but where did it come from? And particularly with incursions, that would be very useful information to collect. And it will also give us a steer on whether the CRB that we have in hand actually has a potential virus susceptibility, yes or no. And the other thing is, if we know more in detail about the CRB origins, it might help us as well with biocontrol agent selection. What biocontrol strain or what biocontrol organism would be the best to use? So when we jump into the virus, we have developed a PCR to detect virus. So that's a plus minus. You either get a PCR amplifies a fragment and you have virus present, or you don't. So what we then do when the virus is present, we take these amplicons and we send them away for sequencing to identify the virus strain. But what we would like to do is go beyond the plus or minus virus to direct virus strain identification. So to develop tool improvement. And the aim for that is so that we can do the virus identification in-house with a short turnaround time. It can be done, if we push it really hard, we can do that within 48 hours with a sequence in hand. And in order to do that, we have generated whole genome sequences of several virus strains. 5 completed, there are more in progress, and it helps us to distinguish the origins and the difference of the different virus strains. And with the whole genome sequence information in hand, we can then go back and develop very targeted assays where we can test for multiple strains, but also from multiple samples that we get from our different partners all at once. So that helps us to monitor the spread and the efficacy of biocontrol virus after release, as well as it will aid us to select the biocontrol agent that we want to employ in a certain region. So from the virus, we can jump into the fungus, the biocontrol fungus, Metarhizium majus. And to your right of this screen, you see a phylogenetic tree with a red circle around Metarhizium majus and the 4 strains that we're currently sequencing as well. Some of this work has been completed, some of it is still in progress. So again, we were using PCR to amplify different different pieces of the genome of Metarhizium majus to then identify the strain via sequencing. And there are the 3 genes that we used so far. Now we actually have the genome sequence of some of the strains, an increasing number of strains in hand. We can again try and get the tool improvements. So we improve the quality of the strains used in the field based on the knowledge that we already have, which ones are the better ones. And we can also, again, once we release a strain in the field, we can follow the establishment and the spread in the field through rapid in-house, quick turnaround, strain identification of field samples. So again, the main target is monitoring the spread and the efficacy of the biocontrol fungus after release. So this is work in progress, partially completed. where do we want to take it next? We'd like to be able to, when we get field samples from our partners, using CRB gut DNA, have a one-step assay to detect the virus as well as the CRB clade. We actually have that in hand at the moment, and we're trying to, we have proof of concept, I should say, of that one-step assay in hand, and we hope to extend it into the real life samples that we get from our partners. And we would like to also do, once we get the plus-minus virus, to also get the follow-up assay for strain identification in a quick turnaround time as well. On the back of that, we would also like to develop a one-step assay for detecting presence of majors and pair that again with not is majors presence, yes or no, and what is the strain? And hopefully we can detect the majors before it actually becomes a visible infection on the beetles. And then eventually, and that'll be a little bit harder to obtain, but hopefully we'll successful in that, is to develop nuclear genome-based assay for CRB. So at the moment we're working with the CO1 gene for mitochondria. We'd like to get a marker or markers in the nuclear genome, which is probably where the genetics for the resistance, susceptibility, tolerance, whatever your definition is, resides. So to get a marker or markers in place that would actually point to that a little bit more conclusively than the mitochondria marker at the moment. And that would point into virus susceptibility. Just for your pleasure, I have put some pictures up on your screen to the top right of a model that is, this particular beetle is lovingly called Billy the Beetle. And this is an actual model that is about 3-4 times the size of a live beetle that can be completely taken apart with all the organs and the bits and pieces inside it, that we are hoping to use for education purposes. That's not something that I'm going to discuss today, but it's such a cute, cool little thing that I thought I would put it up on your screen. And then eventually, finally, my last slide, I would like to thank all the people involved, the usual suspects in the left column there, the people, the key researchers in this particular project, but particularly Nikki Richards, Mitchell Weston, Ella Sawika, Marian Tuboltsev, Rui Jaragui and Sandeep Gupta, who do all the heavy lifting in the molecular, the genetic and the genomic work in this project. Funding comes from New Zealand Ministry of Foreign Affairs and Trade, and there is a whole raft of collaborators that I have to thank, partners in this particular project. And that is me. Happy to answer any questions.
Very good, thank you, Jan. Are there any specific points of interest to answer? Because otherwise I'd like to go to take it through the 3 main areas we've had questions about. Madoka has raised one on estimating the density of beetles, a practical one for the field, and beetles and damage, and that could include Barla's assessment, and there's a little bit to talk about there. We've talked about haplotypes and the use of the haplotype marker for the field monitoring. And then, as well as that, the third tranche there would be the genomic work that's going on. So I propose that we do it in that order. So Madoka, you've written a question about looking at the use of pheromone traps and damage assessment, and can you explain a little more your question?
Yes, thank you. So my question is whether any pheromone trap is now used for the monitoring of the adult density, but always we are looking for the larvae. So larvae density is very higher in everywhere, and so do you think that pheromone trap monitoring is the best way to monitor the CRB density? that's my question to everybody.
I think Mark would be a very good person to answer your question there. Oh, Mark, can you get back to us? He may. He may have some problems, but certainly take it up with Mark, because the PNG Opera have done a large amount pheromone monitoring on a large scale for a long time. And I noticed on the list that some of our Malaysian colleagues are also listening in, and they've got a lot of experience on the pheromone trapping because the oil palm plantation system is a much more stable system than the villages, where we try and monitor as well. So there are complications, but I don't think we're going to get Mark to come in. But at the moment we're working on, with Sulav's working on a review of the pheromone trapping, and together with Mark and others, and we can share that material with you as soon as it comes through.
This is Aubrey.
Aubrey, you know a lot about pheromones.
Yeah, I can comment on pheromone trapping here on Guam. When the beetle first came here, we were told by the pheromone producers that all we had to do was put up a certain number of traps, and we could actually trap out all the adults and problem would be solved. Well, we put up a lot of traps and noticed that trees were getting attacked right in the midst of our mass trapping in the hotel area on Guam. So we forgot that idea, but we continued to have traps throughout the island for surveillance. And sometime later on, I did some mark release recapture experiments. We took trapped beetles, took them back to the lab, fed them up for a couple of weeks and made sure they could fly, and then released these marked beetles within from the center of a large grid of traps. And in these experiments, we always captured much less than 10% of the beetles that we released. So that tells me that our beetles, and we only have the one clade, the CRB they're really not that interested in the commercial pheromone available. And we do have this hypothesis that maybe their, CRBG is looking at a different pheromone naturally, and maybe these pheromones are very good for CRBS, but not for CRBG, but we can't test that because we only have the one biotype here. So that's just one idea. And then the other question is there a good relationship between CRB damage and beetle density? And I don't think anybody's really looked at that relationship. I don't think it's a very strong relationship. So it's very hard to measure density of CRB as adults anyway.
And well, even harder, in some ways it's even harder as larvae. While you might find a lot of them, the distribution is very clubbed, and it is very difficult. Is Ken still there? Perhaps Ken's got a view on the pheromone trapping, because the Hawaiian system has been going for a long time. No, perhaps hasn't heard me. Jeff, did you have a comment?
Yes, regarding rhinoceros beetle density. In the past, as mentioned, one can use damage surveys, but always bearing in mind that a damage survey is there to give an index of the wild beetle population. It's not an actual measure of the beetle population. It's just an index, but that may be all that one can easily get hold of in the field. The rapid damage survey involved counting the number of palms at a certain location which had damage to the recent central fronds, and one could express that as a percentage. And the detailed damage survey involved counting the number of damaged fronds in a significant sample of palms at a particular location and expressing that as a percentage of fronds damaged. So that at least gave some numerical information on in relation to the beetle population at those locations at a particular time. But as I say, it's an index of the population rather than an actual, rather than giving numerical information about the actual population itself.
Yeah, thanks, Jeff. I see a couple of points from Ken. Ken's saying that there's a 25% correlation between trapped catch and density that they measure. So there is a relationship. And also that there is a bias, a sex bias in the trapping. I think usually we've found that about 60% females, 40% males is kind of the average. So certainly more males were more females are caught than males, which is an interesting aspect of their aggregation behavior or their survival. We don't know which. I think it's useful because now people know who's working on the density of the insects and who's interested in the pheromones, and you can cross-link and discuss this even further. We've got another 10 minutes or so before we don't have to stop them, but we will, because it's good to keep to time. But the question of haplotype was raised, and Carmel raised the question of whether haplotypes can mate, can interbreed, and Jeff was also raising the question of the haplotype determination as a determinant of what in regards to the properties of a population. Is there anybody would like to comment on that? Madoka, you've got two hands up. Maybe Sean would like to say something as well.
Yeah, so thank you for lots of comments, and I want to read it now. But what I want to propose is we can unify the method of monitoring in each country, because Guam had a Guam, monitoring way of monitoring. How many days between the monitoring and also collecting the beetles, or how distance between the traps, or that kind of details, information we can share, and then we can have always same way of monitoring, and then we can compare. So that kind of things we need to do it, and I want to propose that we can make the same procedure to monitor. So that is very important in this webinar, and that can be outcome of this webinar. I think that is my proposal.
Yep, very good, Madoka, and I think many of us would be very, very keen on that. To really, we tried last year with SPC to produce the manual of techniques, and the next step is really to standardize those into operating procedures, if you like, and then we can compare one thing directly with another. So we're working on damage. We'll combine on the pheromones and the damage that we're doing, and go from there. Any more comments on the haplotype question?
I was just going to comment, following on from what the speakers have mentioned. I've always wondered how a change in part of the CR1 gene in a mitochondria could give resistance. And from the discussion, both here and in recent literature, it seems that resistance to virus may be due to changes in the nuclear genome rather than some small change in the CR1 gene in the mitochondria. That seems to be what's coming out of literature and the discussion. And I was just wondering if people were coming to a similar conclusion.
Yeah, I think we've always believed that, Jeff. The CO1 gene is a marker of a population. The characteristics of that population are carried in its whole genome.
In the nuclear genome, in other words, yeah.
Yeah, the nuclear plus the mitochondrial genome, because people are finding that there are correlations. Yeah, that's a minor part of it. But what the CO1 gene gives us, the haplotype gives us, is the origin of a population. If that is a single origin, if it's a single haplotype, then probably it carries most of the genes of the population that it came from. And so what we see with the analysis that Sulab presented in the Solomon Islands is most of the invasive populations in the Solomon Islands are a single haplotype. In some areas, they overlap. Where they overlap, it's probable that they've interbred, and where they've interbred, it's probable that they have shared genetic characteristics. The results of that, we don't know yet, but that's where we're moving. The third area, which is really very much related, is the genomics. And as you can see, between us, work that University of Queensland work that's come out of India recently, there is the sequencing is just so much easier than it used to be 2 years ago, 5 years ago, just a dream 10 years ago. And so it's important that that we characterize what we're starting to sequence, so we know what the isolate is, what the beetle is, what the location is, what the population is, before we go through all that process of actually getting a sequence. Because what are we trying to, ultimately we want to relate the sequence back to those characteristics. So I'd just say that that's an important thing, that what we select for sequencing is representative of a population with particular characteristics. But anybody else got any questions or comments on the genomics, where we're going? I'm sure there'll be plenty of, we're seeing papers coming out and plenty to discuss at these meetings as we move forward. Okay.
Yeah.
Well, it looks like we're coming close to the end of our time period. I'd like to thank everybody that volunteered to present and came up and presented and managed their way around all the various buttons for sharing the screens and turning things on and off. And I think it's been a very good and very positive session that we've had together. Mark and Aubrey and I informally keep this thing together, but we'd welcome any suggestions on how we run it differently, what issues we should put into it. But it does give us a forum to discuss and interchange. And I'm very pleased that we've had quite a few people from the countries that are affected listening to the webinar and also presenting. And I'd like to make special mention to Bala. Bala hasn't done many presentations before, and I thought her presentation was excellent. And at the other end, thank you very much, Jeff. You've presented many times before and have a vast amount of knowledge on CRB. So thank you very much for sharing it with us this time. And also, of course, this has gone very smoothly. So I'd like to thank University of Guam and Brian for putting it all together for us. And so, yeah, all I can say is keep well, keep out of the way of COVID. Christmas is coming, enjoy it, and we'll keep in touch, and we'll catch up with you all again with another one of these in the new year. Okay, thanks very much.